honour to be delivering the Lupski Lecture for 2022. Jim Lupski was the chair of our scientific advisory board for the study, so we're indebted to him for the wisdom and energy that he brought to that role. So I'm speaking today on behalf of a huge team of people. There's the core team of scientists and bioinformaticians at the Wellcome Sanger Institute that you can see assembled on the helical staircase. There were the 200 consultant clinical geneticists, 100 laboratory scientists, 50 research nurses and genetic counsellors in the participating regional genetic services, the thousands of patients and their families and the support groups, and the DDD management committee. And the five members of our current committee have all been with the project since the very beginning, meeting one day a month to steer the project. So let's think back to what was happening in the world in 2010. So it was the year of the Haiti earthquake. Some of the older members of the audience might recall the volcanic eruption in Iceland that grounded planes in Europe for a couple of weeks due to the ash cloud. And some of the younger members might be aware that Instagram launched in July of 2010. So what was happening in the world of genomics at that time? So the 1,000 Genomes project had just been, the pilot for that had just been published. And that was the whole genomes of 179 individuals and the whole exomes of about 700, and contained two trio whole genome studies. And that was the cutting edge, that was the state of the art for genome-wide sequencing internationally at the time. And if you look across to the PubMed graph, you can see that there were literally just a handful of papers mentioning whole exome sequencing in PubMed in 2010. And this is the date DDD started. And Array CGH was just transitioning from research into the clinic. And at the time the project started, the majority of the centres contributing had been able to run a genomic array on the patients prior to recruitment. So what were the drivers and enablers of DDD? I think the first was the choice of congenital disorders or severe developmental disorders of early childhood as the group that we would study in the project. We chose those because there was minimal population data available in 2010. So as well as pre-Instagram, this was the pre-EXAC, pre-Nomad era. EXAC didn't come along until 2014. So we needed to have conditions that would have a strong signal, a distinctive phenotype, highly penetrant Mendelian disorders with a high de novo mutation burden, and where there was a minimal environmental contribution. It was the rapidly developing and evolving technologies for whole exome and whole genome sequencing, and a desire and opportunity to match the world-class genomics at the Sanger Institute with high-quality phenomics in the NHS genetic services. There was also a huge diagnostic need. So sitting in clinic, I could diagnose only about a quarter of the patients that I saw. At the Wellcome Sanger Institute, we had access to amazing technology by a team who'd delivered a huge proportion of the Human Genome Project. And we had Decipher, which was a web-based platform linking phenotype and genotype, which we'd established in 2004, and which was widely used in the genetic services for the interpretation of array results, and was able to provide the informatics infrastructure for the project. So right from the start, the project had twin aims: diagnosis, transforming the diagnosis of rare disease in the NHS, and discovering the genomic architecture of developmental disorders. And that was reflected in the structure of the project, in the partnership between the 23 NHS regional genetic services and the Sanger Institute, and in the funding which came jointly from the Department of Health and Wellcome. And our plan was to collect over 30,000 DNA samples from more than 10,000 UK families with a young family member affected by rare disease, severe developmental disorder, where it had not been possible to identify a diagnosis using NHS testing. So in a project of this scale, you need to develop an identity, a sort of branding, and the Wellcome Trust Case Control Consortium were just starting to publish the big GWAS studies. And so it seemed that DDD might be a good next step. And so we coined the term Deciphering Developmental Disorders. My daughter, Sarah, kindly sketched this lovely logo for us, and the graphic artists at Sanger converted that into a professional quality image. And we worked with companies like Origin to develop custom packs for saliva sampling, because the adult spit packs weren't suitable for young children. We needed a more individualized sample collection system. And here you can see it with the DDD icon. We ordered tens of thousands of these. So building the collaboration, it was a real partnership. We wanted to build trust, encourage participation, ensure that everyone understood their roles and responsibilities. We wanted people to be collaborators, not just recruiters. So we wanted full engagement with DDD and a commitment to manage results and deliver them to patients. And we provide opportunities for research, but we wanted to ensure an obligation to variant annotation. And we wanted to work as a distributed network of expertise to advance diagnosis and discovery, whilst retaining individualized care close to home for patients and their families. So in a project of this scale, you need excellent communications, and we were enormously fortunate in appointing Caroline Wright as our project manager, who coordinated an amazing communication strategy with bi-monthly newsletters, with a website, and with interfacing with all the teams that were needed to support a project of this complexity, particularly the Decipher team that needed to develop new informatics to support recruitment, and the visualization of SNVs. So the genomic side of the study was focused on the data generation, sequencing 33,500 exomes, 12,500 SNP arrays, over 5,000 exon level CGH arrays, and all in all from 10,000 trios and 3,500 singletons. But right from the start, we wanted to match that world-class genomics with first-class phenomics. So we collected HPO terms, family history, pregnancy history, morphometry, milestones, and images through the Decipher portal. I set up a workshop, for a week-long workshop with Peter Robinson, to refine the HPO, to prune out redundancy and fill in gaps, so that we had a menu of codes that we could use to codify phenotypes across all body systems, ready for the launch of the recruitment. And our data analysis, alongside our twin aims, our data analysis was also in two halves. Firstly, a quasi-clinical reporting against a panel of 2,000 known developmental disorder genes, with the candidate variants fed back to the local service for clinically and laboratory evaluation. And then a research analysis where we focused on trying to identify new causes of developmental disorders. by looking at genes with an excess of mutations in patients sharing similar clinical features. So this was the clinical filtering plan for managing the huge number of variants coming from an exome analysis, stepping down from the 20,000 to the 400 rare functional variants, filtering those against the DDD G2P gene set, and coming down finally to not just a handful of likely diagnostic variants. And in order to achieve that, we had to build this gene panel, and this was led by David Fitzpatrick with help from me. We built a set of gene disease associations which specified locus, genotype, mechanism, disease entity, and the evidence behind that, which could be used in a computerized setup to facilitate this filtering process. And then the candidate variants, in the way that the phenotype had been added at recruitment by the centres, the candidate variants were fed back through Decipher. And this was the report of our analysis of the first 1,000 patients in the study, Caroline's paper in the Lancet. And you can see this is a Circos plot from chromosome 1 round to 22, X and Y. And then the phenotypes, ID, seizures, congenital heart disease, autism spectrum, hearing impairment, cleft, scoliosis, etc. And these are the genes with their locations on the chromosomes where variants were found, and these were the phenotypes that they were associated with. And there are two key messages that came from this. The first, that the underlying biological complexity that we were dealing with, and secondly, the fact that this was doable. This worked. You could do genome-wide sequencing at scale and deliver diagnoses and identify new genes. So that was encouraging. So Matt Hells has led the scientific strategy for the project, and a lot of that has been focused on burden analyses, and particularly looking for an excess of de novo variants in genes over and above that which would be expected by chance. And so here's a Manhattan plot across all the chromosomes with a threshold for genome-wide significance, which was established by Jeff Barrett, the statistical geneticist on the management committee at the start of the project. And you can see about 100 genes rising above this threshold, genes which are statistically associated with developmental disorders. And you can see quite a lot of familiar names across the top, but in fact, in 2015, these weren't familiar names. ARID1B, DDX, 3X were new conditions discovered through the study. And really emphasized the importance of data sharing in driving discovery. So here were patients with de novo variants in PPP2R1A in Glasgow, Newcastle, and Belfast. If those patients' data had remained within those individual centres, it would never have been matched with the others, and we'd never have been able to identify PPP2R1A as a new cause for a distinctive developmental disorder. Similarly with PCGF2 in Newcastle and Exeter, COL4A3BP in Sheffield, Birmingham, and Exeter, and so on. So I've shown you the data from the first 1,000 patients, but Caroline led an analysis looking back again at that data 3 years on. So in 2014, we'd been able to achieve a diagnostic rate of 28%, but using that same data in 2017, we were able to lift that to 40%. And a third of that uplift was due to new genes we discovered within DDD, a third due to genes others had discovered, both types of which have been entered to the new DDG2P, and a third due to tweaks to our analysis pipeline and new analysis approaches. So driving a project like this requires a lot of engagement, and we visited every site at the start to help them set up their recruitment. But halfway through, Caroline organised a bike ride, and here are Matt and I about to cycle from Great Ormond Street Hospital in London to Hingston. We needed to engage research teams, and we brought the collaborating centres representatives on site annually for a big meeting to update them on the progress in the project. And we worked very closely with support groups, Swan UK supporting the undiagnosed families in the project, and Unique, the newly diagnosed. And we asked Unique to set up single gene trifold leaflets for the new conditions that we were discovering, so that families, when they were seen in clinic, were offered something more than just an acronym. They actually had something that outlined what were the key clinical features of the conditions, and Unique did a brilliant job weaving the medical facts with photos and family stories to make these leaflets. So they're now over 70 of these leaflets, and if you feel you could help to write one, please do contact us or contact Unique. They really appreciate that. These leaflets have been hugely popular with families. But the main vehicle for communicating our results was through publication, and here are some of our major publications. So the importance of de novo mutation in the etiology of developmental disorders, the value of collaborative work in identifying new disease genes, the contribution of recessive genes to developmental disorders in the UK, and showing that this was a scalable approach, and this paper by Patrick Short on de novo mutations in regulatory elements enabled by the extended exome that we had deployed in the study. So here is a column in the middle for representing developmental disorders, and showing that the biggest component by far is caused by variants in monoallelic genes in the coding sequence. Next come a contribution from splice variants, and this analysis was led by Jenny Lord, and then non-coding regulatory elements, Nicola Whiffin doing analysis of UTR variants, particularly in MEF2C, and Patrick Short. And Hilary Martin has led big analysis of the recessive contribution to the DDD study, both autosomal recessive and X-linked recessive genes. And most recently, Ruth Eberhardt has led an analysis of mosaic chromosome alterations in children with severe developmental disorders recruited in the study, and they contribute about 0.5% of the diagnoses to the study, and importantly, often found only in saliva and not in blood. So do listen to Ruth's talk later in this conference. So the scientific strategy for DDD has been led by Professor Matthew Hurls, Head of Human Genetics at the Sanger Institute. It's been focused very much on new mutation as drivers of developmental disorders. And here's a little cartoon of a gene with the coding sequence in the exons shown in green, little regulatory elements in orange. And you can see that the mutational target of the coding sequence is much larger than that of the regulatory elements, which tend to be have quite a small footprint in the genome. And this gene has just an average of, shows 3 exons, but in fact genes generally have 10, 20, 30 or even more exons. And so the chance of the mutation landing in the coding sequence is much higher than hitting a regulatory element. In addition, when a mutation does land in the coding in the sequence, it's much easier to impair or lose function than it is for the mutation to enhance or alter function. And so the opportunity for gain of function, again, is much rarer, and you need much larger numbers to try to address those discoveries. So in addition, the phenotypic consequence of mutating a regulatory element may be subtly or substantially different to a loss of function variant hitting the coding sequence. It might, for example, alter the tissue specificity or the time in embryonic development when that regulatory element is active. So ways to improve the power of detection. We've shown that having a large data set, 13,500 families, has enabled a lot of gene discovery, but in order to build discovery of rarer or smaller disease genes or new genomic elements, we need bigger and bigger data sets, and MAPS leading collaborations with Genomics England, NIMEG and GeneDx and other partners to try to increase the power to find these harder to find elements. So looking at our column here, we're left wondering what's going on for all the patients in the top half of the study where we can't currently find a diagnosis. And this is about 50% of the cohort. The phenotypic burden is pretty equivalent to the diagnosed cases. So how do we find these additional causes of developmental disorders? So Lizzie Radford's leading a project on saturation mutagenesis, and she'll be talking about her work with DDX3X later in this conference. Katrina Andrews is devising algorithms to try to look for altered function variants and genes that work through that mechanism to create or cause developmental disorders. And she'll be giving a presentation later in the program. And Patrick Campbell's been analysing the factors that affect the likelihood of finding a diagnosis in DDD, and do visit his poster. So the ACMG variant classification rules have been very impactful in clinical practice in the implementation of genomic medicine. And here's a snapshot of these rules with which I'm sure you're all very familiar. And then there was a sort of Bayesian look at these rules that was led by Sean Tavtipigian, that enabled a sort of more quantitative scale, which is particularly helpful given the sort of huge number of variants that fall in the sort of uncertain range in this scale. So we've got nearly 20,000 variants returned by DDD, and it's been brilliant that 83% of them are annotated as pathogenic or benign or uncertain by the contributing centres, but that still leaves us with several thousand variants without an annotation. So we looked at what we could code of the ACMG criteria using the data available to us. And this is work that's been led by Caroline and David and the Decipher team. And here's a plot showing the ACMG scale from benign to pathogenic, and then the way that the variants have been called by the clinical teams: benign, likely pathogenic, pathogenic, uncertain, and the unannotated variants. And we're focusing on discrepancies and also on the uncertain or unannotated variants where it suggests from the coding that we've done that these variants may be disease causing. But we can also dive into the discrepancies where there are variants that appear to score strongly for pathogenicity, but have actually been annotated as benign, etc. There's a lot to be learned from exploring the discrepancies in this sort of representation, and we're only just at the start of that effort. So phenomics have always been a core part of DDD, and because of the rich phenotype that we've managed to acquire on every participant in the study, we've been able to write little disease models for many of the disease genes. So here's CDK13, 12 patients with pathogenic or likely pathogenic variants, and these are those growth and development parameters. And you can see here that on average, these children are slightly shorter, with the average height being around -2 standard deviations. And this computational analysis has been led very much by David Fitzpatrick, working up in Edinburgh. And the idea is that with the categorical data, all the HPO terms we collected together with this quantitative data, we could merge that together to derive a phenomic disease model. And then we could apply that in conjunction with the variant and the ACMG variant classification, which you can see here as 0.8, so well in the uncertain category. But by building in the phenome model, which is very strongly in favor of CHD7 for this child with coloboma and dysplastic ears, you can then reach a confident diagnosis of Charge syndrome for this patient. So DDDs use Decipher throughout, and here's a map showing the first and last, the locations of the first and last authors in our publications, the first 125 publications from the project. And you can see that although a lot of that's in the UK, that the project has actually reached out and collaborated very much internationally, and now reached 262 publications. Throughout the world, centile charts are a standard of care in looking after young children to monitor their development and growth. Children with developmental disorders are ill-served by these charts. And here in Decipher, we've been able to develop these individualized centile charts based on children with likely pathogenic or pathogenic variants, so that for many of these disease genes now we can produce customized centile charts, so that a child here with a head circumference of -4 standard deviations is growing entirely normally for a child with EP300. But that would be a great cause of concern if it was plotted on a conventional chart. There are also increasingly new therapies coming on the horizon for rare disease. So here's a paper published in January 22 on 2 infants treated with PIK3CA-related overgrowth spectrum by a drug called alpelisib. And here you can see an absolute dramatic improvement in the resolution of the overgrowth with this drug. And through Decipher, we are able to search for de novo mosaic variants in PIK3CA, find these 3 patients in addition to the patients in DDD, and send a communication in a Decipher clinical alert to their clinicians to notify them about this paper, which is directly relevant to this very small subset of patients. So in DDD, we've now sequenced 33,500 individuals with data on 10,000 trios, found diagnoses for over 5,000 children, got a complementary analysis proposal, so 350 projects run in the recruiting centres in partnership with their local universities, mainly on genotype-phenotype correlation or on new gene discovery. The DDD dataset's been used to devise new algorithms like Splice AI. More than 1,000 children in the project received a diagnosis as a direct result of a gene we've discovered in DDD. And we're continuing to deliver new diagnoses. Over 100 diagnoses have been returned this year. And hoping to show, make genomic medicine work to deliver improved healthcare. So in summary, we've discovered 67 completely new disorders in 11 years. You'll notice I've built an extra block on the child's tower, and that's very much an invitation to you to use Decipher to make your patients discoverable, to enable their recruitment to research and therapeutic trials by sharing their variant and phenotype, and to help us build these data models, which will then enable more accurate diagnosis and better patient management. And with that, I'd like to thank you very much for listening. Thank everyone who's participated to make this project such a success. Thank you.